So the fundus and the body mucosal cells are going to be producing most of the gastric juice. We have three types of mucosal gastric cells. First one is going to be the parietal cells, and the parietal cells are going to be responsible for the production of the hydrochloric acid and the intrinsic factor. Hydrochloric acid is essential as it will be denaturating the proteins, and this is the first step in the protein digestion. Another thing is that it activates the pepsinogen into pepsin. And the third major function here of the hydrochloric acid released by the parietal cells is that it kills the bacteria that we're going to be ingesting. Another thing that will be produced by the parietal cells is going to be the intrinsic factor. And the intrinsic factor is a glycoprotein that is required for you to be able to absorb vitamin B12 from the small intestine. So you see on here, traveling down from the stomach to the small intestine, we're going to have the food on here, and it contains vitamin B12. In order for me to conduct vitamin B12 from my small intestinal cells to my blood, I need to have intrinsic factor. And the intrinsic factor is going to be released up here from my stomach. And what cell exactly would be releasing the intrinsic factor is going to be your parietal cells. Having deficiency of vitamin B12, this will result in a type of anemia that we call the pernicious anemia. Pernicious anemia is a type of anemia that results due to deficiency in vitamin B12. Another type of mucosal gastric cells we're going to have is chief cells. Chief cells are going to be releasing the main enzyme responsible for digestion here in the stomach. This is the pepsin, and it's going to be released in an inactive form, which is the pepsinogen. The pepsinogen is going to be activated by the hydrochloric acid. So if we see on here this reaction, we start with pepsinogen released from the chief cells. In the presence of hydrochloric acid, you're going to be getting the active form, which is the pepsin. Pepsin is the enzyme that's going to be breaking down the protein. Also, if you see on here, the pepsin itself going to be stimulating the conversion of more pepsinogen to get more of itself. So you see two factors here will activate the pepsinogen into pepsin, the hydrochloric acid, as well as the pepsin itself. Another cell that we're going to have in the gastric mucosa, this will be the enteroendocrine cells. And from its name, entero means related to the gastrointestinal tract, and endocrine means I am releasing hormones in the bloodstream. I am releasing chemical messages in the bloodstream. So what chemical messages are you sending out from those cells of the stomach? We are releasing many hormones like the gastrin, for example. The gastrin is a hormone that will be released that will be stimulating the stomach to contract, allowing propulsion to take place, as well as increasing the glandular secretions for you to release more gastric enzymes to help with the digestion. So again, two effects here of the gastrin. First one is to stimulate and increase the motility and stimulates the gastric secretions. we're looking on here on this diagram, three types of gastric mucosal cells that we can see. A type of cells that will be releasing the pepsinogen. This is going to be my chief cells. A type of cells that would be releasing the hydrochloric acid as well as the intrinsic factor. This is going to be my parietal cells. And the third type of cells that we can see in the gastric mucosa, this is going to be the type of cells releasing the hormones, and this is called my enteroendocrine cells. Moving on to the gastric contractile activity. The stomach is like your heart. It generates its own electrical impulses. If you remember back when we discussed the heart, the pacemaker of the heart, this is what we've called the SA node, and this is where you generate the normal electrical impulses that will travel to the rest of the heart, causing the heart to contract. Here is the same concept when we deal with the stomach. The stomach also has its own pacemaker, and the pacemaker cells of the stomach, those are the cells of Kagel. Cells of Kagel are going to be acting as the pacemakers that will create the basic electrical rhythm, and this basic electrical rhythm will cause the generation of the peristaltic waves, the contraction waves that will be moving towards the pylorus, and we're going to have 3 peristaltic waves per minute. What stimulates the Kagel cells to depolarize at a faster rate are two major stimuli. Those are gastrin as well as the distension of the stomach. So two major factors here that I will see stimulating the Kagel cells to increase the rate of depolarization. Those are the distension of the wall of the stomach. So if I have distension of the wall of the stomach, this directly tells me that you've got food inside. And if you've got food inside, you need to stimulate the contraction for you to move this food from your stomach to the small intestine. Another thing is going to be the gastrin that we did just release from the enteroendocrine cells. So whenever we get food inside the stomach, this will activate the enteroendocrine cells to release gastrin, and the gastrin, one of its effects is going to be to stimulate the gastric secretions as well as the Kagel cells to depolarize at a faster rate, increasing the motility of the stomach. Most vigorous contractions will be taking place towards the pylorus. So if we're looking on here at the contraction waves taking place in the stomach, we can see that I have a propulsion movement. So the food is going to be moving down like this. And the most vigorous contractions that will be taking place, those are going to be taking place in the pylorus, the pyloric canal, and this is where the grinding is going to be taking place. I have very vigorous muscular contractions taking place on here. This is where I will be performing the mechanical digestion now. You see, only a small part of the food is going to be moving down to the duodenum, but most of the food is going to be moving back into the stomach. Only smaller amounts of the chyme will be moving to the duodenum, and most of the food is going to be moving backward, and this is what we're going to call the retro propulsion. Getting more grinding, more mechanical digestion, more chemical digestion to this amount of food that's going to remain in my stomach. So you see on here, all this amount of food will be broken down another time, passing through the pylorus where I have the most vigorous muscle contractions. You see on here, lesser curvature is going to be attached to the lesser omentum. Lesser curvature of the stomach is going to be attached by the lesser omentum. Checking this question out, what are the digestive processes of the stomach? Do we perform any chemical digestion? What do you think? What do you think? Do I perform any chemical digestion? Yes. Yes or no? Yes. So can you remind me what's the name of the enzyme that we're going to be releasing, and what's the cells that would be releasing the enzyme responsible for the chemical digestion inside the stomach? We are performing chemical digestion in the stomach under the effect of the pepsin. And the pepsin here is going to be released in an inactive form. This is my pepsinogen. And what is the cell that would be releasing the pepsinogen? This is going to be my exactly chief cell. Do we perform any ingestion through the stomach? Do you normally eat directly into your stomach? Through your stomach? What do you think? Do you have any ingestion? No, ingestion takes place in the mouth, exactly. So no ingestion will be taking place. Do we allow any propulsion in the stomach? Yes, we do. And where gonna be the most vigorous muscular contractions, the grinding movements that we're gonna be performing in the stomach takes place in the pylorus. And can you remind me, what is the pacemaker of the stomach muscular contractions? Those are the cells of Kagel. What do you think? Do I perform any mechanical digestion? Yes, grinding movement takes place where again? Again is close to the pylorus. So yes, I perform a mechanical digestion on here. So what's going to be the good answer on here is going to be A, C, and D. Moving on to the next part of the gastrointestinal tract, this is going to be the small intestine, and the very proximal part of the small intestine that would be receiving the food from the stomach. This is going to be the duodenum. Duodenum is the shortest of the segments of the small intestine. It's going to be receiving the bile secretion as well as the pancreatic secretions from the liver and the pancreas. And those two ducts, the bile duct coming from the liver and the main pancreatic duct coming from the pancreas, will fuse together, and they will be forming what we call the hepatopancreatic ampulla. And the hepatopancreatic ampulla is what will conduct the liver and the pancreatic secretions to the proximal part of the small intestine, my duodenum. It enters the duodenum as a major duodenal papilla. Both are going to be controlled by a sphincter that will be surrounding the hepatopancreatic ampulla. So we're going to have the sphincter between the liver. What do we call something related to the liver is hepa. So hepato here, hepato is related to the liver and related to the pancreas. It's going to be pancreatic, and it's a sphincter that will be regulating the conduction of the fluids on here. This is going to be my sphincter. So the sphincter here guarding the hepatopancreatic ampulla is going to be called the hepatopancreatic sphincter. So if we're looking on here, just moving to the slide to show you how the duodenum will be receiving the bile and the pancreatic secretions. You see, the liver is going to be producing the bile, and this bile is going to be conducted into two ducts on here. Those are called the right and left hepatic ducts. Both hepatic ducts will conduct the bile into a common hepatic duct. The common hepatic duct will be allowing the bile to travel back up to a muscular sac on here that will be located on the inferior surface of the liver. This is called the gallbladder. And the gallbladder function, as we're going to learn, it's going to be to absorb the fluid out of the bile and make it concentrated. This concentrated bile will then be released from the gallbladder to the cystic duct and then to the common bile duct. The common bile duct will be conducting the bile down, and it will be fusing with the main pancreatic duct carrying the pancreatic secretions. They will form together my hepato and pancreatic ampulla. So hepatopancreatic ampulla is simply the duct that will be receiving the secretions from the liver and from the pancreas. You see on here, this elevated mucosal ridge is my major duodenal papilla, and the duct on here, the ampulla, is going to be guarded by a sphincter. So what we're going to call the sphincter on here, guarding the conduction of the bile and the pancreatic juice, this is going to be the hepatopancreatic sphincter. So again, the pathway on here and how the pancreas and liver are going to be related to the duodenum. Right and left hepatic ducts will be conducting the bile to the common hepatic duct. Common hepatic duct will conduct the bile to the gallbladder. This is where the gallbladder will be concentrating the bile, waiting for the food to come down here into my duodenum. After you get food down into the duodenum, or in the same time, I will be contracting the gallbladder to release the bile through the cystic duct, the duct of the cyst, or the duct of the bladder, cystic duct. The cystic duct will conduct the bile down to the common bile duct. Common bile duct with the main pancreatic duct will form the hepatopancreatic ampulla. Hepatopancreatic ampulla will be guarded by a sphincter. We call the sphincter on here the hepatopancreatic sphincter. Right, this is one of the most important diagrams in this chapter. Highly important. We're going to see it another time when we check the atlas in a second. You see on here, the next part of the small intestine is going to be my jejunum, and the jejunum is going to be the middle segment of the small intestine, and it's about 8 feet in length. Next part, and the most distal segment of the small intestine, which is almost 12 feet in length, this is going to be the ileum. And if you remember back from our very first session, from the very first slides in this chapter, we did mention that most of the absorption will be taking place in the small intestine. And remember, what if there will be part of this food that you failed to absorb or you failed to break down? What you're going to do with this remaining part of the food is going to be conducted to the large intestine to form the feces that will be released to the outside. So you see on here, what part of the small intestine will be conducted, the unabsorbed, undigested part of the food, to your large intestine is going to be the most distal part, which is my ileum. So you see on here, again, the shortest segment of the small intestine is the duodenum. It receives secretions from the pancreas and the liver. Next is going to be the jejunum, and finally we're going to have the ileum that will be connected to the cecum by the ileocecal valve. Why do you think I will need a valve here to regulate the movement of this part of the food that I failed to digest from the ileum to the cecum, which is this pouch-like structure of the large intestine? Why do you think you will need a valve? Simply to allow a unidirectional movement. I don't want to have stools moving back up into my small intestine. I want only the stools or the remaining part of the food that was not digested and absorbed to pass from the small intestine to the large intestine and not to move backward. This is very, very important to pay attention to. We're going to have a valve on here. Moving on to the different functions of the small intestine. First set of functions in the small intestine, those going to be the chemical digestion. And if you remember, chemical digestion means I am changing the chemical structure of my food. What would be responsible for the chemical digestion in the small intestine? We've got secretions from the liver. What is the main secretions that you're going to be receiving from the liver is going to be, the bile, exactly. And the bile will be responsible to emulsify the fat. What I mean by emulsification of the fat. So if you have oil and you put this oil in a cup of water, like this. So this is water. Would you be able to mix the oil with water? What do you think? Will you be able to mix them together? Actually, they will separate. So the oil will keep floating at the top of the water and it will not be mixed with it. So how do you think I will be able to break down fats if the fat is insoluble? Is water insoluble? So how would I be able to break it down if I have the enzymes which are breaking down the fat located inside the water? I need first to make it more soluble. So rather than having a huge fat particle like this and my enzymes are only getting exposed to the peripheral parts, how about breaking it down into smaller pieces? So I would break down the fat into much smaller pieces like this, so I get a much higher exposure to the digestive enzymes breaking down the fat. Is the emulsification a part of a chemical digestion? What do you think? Yes, I did reduce the size of the particles for me to have much better exposure to the enzymes, so it's going to be the first step which is needed in the fat digestion. And this will tell you why people who will have the gallbladder removed, they will not be able to concentrate the bile. And if I'm not able to concentrate the bile, would I be able to emulsify the fat? So the fat will not be emulsified. So what's going to happen to those fat particles? What do you think? They will remain large in size. So large part, the core of the fat, which has not been exposed to my enzymes, because the enzymes are only getting exposed to the peripheral side of the fat particle. So all the core of the fat particle will be kept unbroken because it was not exposed to the enzymes. And if you did not break down the fat, who will break it down for you? Who lives in the intestine and will break it down for you is going to be the bacteria. So the bacteria will come and will break down this fat, and that's why people will develop gases in the gastrointestinal tract, and they would have other complications, like for example the fat-soluble vitamin deficiency, because I failed to break down the fat, and if I didn't break down the fat, I'm not able to extract the fat-soluble vitamins, and now if I'm not able to extract those fat-soluble vitamins, I will be deficient in those vitamins. Another factors that will help with the chemical digestion taking place in the small intestine is going to be the pancreatic secretions. Pancreatic secretions, which were conducted through the main pancreatic duct, and we have many enzymes on here that will be released from the pancreas, like the trypsinogen, like the procarboxypeptidase, like the chymotrypsinogen, the amylase, the pancreatic amylase, the pancreatic lipase, and the nucleases. All those are digestive enzymes that will be part of the pancreatic secretions. So pancreas would be responsible for digestion of proteins, and the proteins here would be digested by the procarboxypeptidases, trypsinogen, and the chymotrypsinogen. It will play a role also in the digestion of the proteins and the carbs. The carbs. Carbs going to be broken down by the amylase enzyme. Fat's going to be broken down by the lipases, and the DNA and RNAs that we ingest will be broken down by the nucleases. Alright, so again, pancreatic enzymes will help with the breakdown of all the nutrients. Another factors that will help with the chemical digestion taking place in the small intestine is going to be the secretions from the small intestinal cells. Small intestinal cells, or what we call the brush border enzymes, those are like my enteropeptidases, secretin, and we also going to be releasing some hormones like the cholecystokinin. Other functions of the small intestine, it would be the absorption, and two factors will be helping here with the absorption taking place in the small intestine. In order for me to absorb, I need to have a maximal surface area for this absorption to take place. So you see on here, the mucosa is gonna have folds, and I will have the arrangement of the mucosa lining in the form of villi. Also, those mucosal cells at the top of those villi will have microvilli. So if you are magnifying one of those cells on here, how this cell will look like? The cell will have a cytoplasmic membrane with those finger-like projections coming out. Those are my microvilli. Don't get confused between the microvilli, which are at the top of the mucosal cells, and the villi, which are the finger-like projections of the mucosa lining itself. So villi are formed by mucosa, and the microvilli are formed by the cytoplasmic membrane of the mucosal cells. A third function of the small intestine is to allow the propulsion. And if you remember, two types of movements that I can perform in the small intestine. I have the propulsion taking place by contracting the longitudinal muscle layer. This is called my peristalsis. And if you remember, when you contract the constrictor muscles, the circular muscle layer, you're gonna be performing segmentation. And remember, segmentation is a form of a mechanical digestion. So please add on here to your notes, propulsion / mechanical digestion. So propulsion, movement, and this is taking place mainly by peristalsis. And the mechanical digestion is going to be mainly taking place by segmentation. You see on here, this is how the mucosal lining of the small intestine is going to be arranged. The mucosal lining is going to be arranged in the form of those finger-like projections coming out and pits going down. Those are the villi. The microvilli are this brush border of the cells. If you see on here, the mucosal lining will have the three different types of vessels moving in and out. So we're going to have arterial blood vessels, arteries. We're going to have venules. We're going to have lymphatic vessels. All the three different types of vessels will be pathing in those finger-like projections of the mucosa. Again, what is the layer that will be located just below the epithelium? Here, any kind of epithelium is going to need to be supported by connective tissue. This is my lamina propria, if you remember, and the type of connective tissue, this was my areolar connective tissue, which allows the flow of the fluids carrying the nutrients and oxygen to the epithelium. If you remember, epithelium is always avascular.
Alright, so let's move on to the anatomical structure of the different parts of the lower gastrointestinal tract. If you see on here, the esophagus, which is part of the upper gastrointestinal tract, will conduct the food down to the stomach. If you see on here, the stomach is going to be divided into different regions. First region that will be surrounding the entrance of the esophagus, this would be the cardiac region, and we're going to call this sphincter located in the cardiac region, this is going to be the cardiac sphincter. This is my cardia on here. I have this dome-shaped structure, dome-shaped part of the stomach. This is the fundus of the stomach. The stomach will be divided, you see on here, the mid portion of it. This is what we're going to call the body of the stomach. And we have the pylorus. And if we check here, what guards the pylorus, what guards the movement of the food through the pyloric canal to move to the proximal part of the small intestine, is a sphincter located at the end of the pylorus. This is going to be the pyloric sphincter, which is going to be regulating the movement of the food from the stomach to the small intestine. Also, what we can see on here, we can find two curvatures of the stomach. We have a large curvature and we have a smaller one. So we're going to call the large curvature on here, this is my greater curvature. Compared to the small ones, this would be my lesser curvature. If we see on here, this C-shaped part of the small intestine, which is the proximal and the shortest part of the small intestine, which is going to be receiving the food from the stomach, this is called the duodenum. The duodenum will conduct the food here to the next part of the small intestine. This is the duodenum, and the duodenum will conduct the food here for you to complete the digestion and the absorption to the distal part of the small intestine. This would be the ileum. If you see on here, the ileum will be conducting the remaining part of the food that I failed to digest and I failed to absorb to the large intestine, to part of the large intestine that would be like a pouch-like structure. This is called the cecum. So we're gonna call the junction between this part of the small intestine, which is the ileum, and this part of the large intestine, which is the cecum. So we're gonna call the junction between the ileum and the cecum the ileocecal valve. Why is it a valve? Why do you need a valve on here? Because you simply want the part that you did not digest from the food to pass from the small intestine to the large intestine, and not to get feces back from the large intestine to the small intestine. All right. So again, 3 parts on here that we can see for the small intestine. We have this C-shaped structure. This is the duodenum. Then we're gonna have the jejunum, and finally we're gonna have the ileum, and the ileum will conduct here the part of the food that I failed to digest and I failed to absorb to this pouch-like structure of the large intestine. Again, this is what we call the cecum. If you see on here, I have a worm-like structure. This is the vermiform appendix. Vermiform appendix is gonna be attached here to the cecum. What's gonna happen to the part of the food that you did not break down or you failed to absorb? This will be forming the feces, and the feces will be conducted up in here from this region of the large intestine. This is my cecum, to the part of the large intestine that would be going up. This is my ascending colon, and then the ascending colon will conduct the feces to the transverse colon, to the descending colon, and then we're gonna have this S-shaped structure, this S-shaped part of my large intestine. This is the sigmoid colon, and then from the sigmoid colon the feces will be conducted into a straight part of the colon. Straight is recta. So we're gonna call the straight part here of the large intestines, this is the rectum. And the rectum will be conducting on here the feces to travel to the outside through the anal canal, and its opening is going to be the anus. So you see on here, started with the movement of the food from the oral cavity to the pharynx, and then from the pharynx to the esophagus, and then from the esophagus to the stomach. We mentioned the different parts of the stomach on here, the C-shaped structure of the small intestines, the proximal part of the small intestines. This is my duodenum. Then I will pass through the jejunum, and finally the distal part of the small intestine is my ileum. Again, the ileum will conduct the undigested and absorbed part of the food to the cecum through the ileocecal valve. I will be moving the feces up through the ascending colon to the transverse colon, then to the descending colon, to the sigmoid colon, and finally to the straight part of the colon. This is the rectum, and the rectum will conduct the feces to be removed to the outside through the anal canal, which is going to be guarded by 2 sphincters, as we're going to discuss in the defecation.